And in this video, I've decided to cover a really interesting topic of genetics, which is cloning. So we are going to understand the concepts of how cloning occurs. So before that, you need to know what is a clone. A clone is basically a group of genetically identical organisms That means their gene pool is exactly the same alleles, same gene combinations, everything. Or it is a group of cells derived from a single parent cell, in which case also it's going to share the genetic information with all of those cells from a single parent cell. So this process of cloning, it occurs through a process known as a somatic cell nuclear transfer, SCNT. So it is a method of reproductive cloning that we are about to study in this video. So somatic cells are basically those cells that are not the sex cells or the gametes. So all the other cells in our body apart from the gametes are known as the somatic cells. So let's go ahead and study the different parts of this entire process. So step 1 is going to include the following process. A female animal, and in this example let's take a sheep. It will be treated with hormones. For example, the follicle stimulating hormone to produce more eggs, and this will stimulate the development of eggs. The second step after that stimulation is the nucleus from an egg cell. Now very, very carefully you have to understand this. The nucleus from the egg cell is taken out. It is removed. I will also explain this entire process diagrammatically, but right now you need to concentrate on the steps. So first of all, we stimulated egg development in a female animal, and then we took an egg cell and removed the nucleus from it. So basically what did we do is we removed the genetic information away from that egg cell. So when we remove the nucleus, we have basically removed the genetic information of the egg cell. The third step that we are going to do is the egg cell, without the nucleus now because it was removed in the second step, egg cell is fused with the nucleus from a somatic body cell. Now usually the egg cell is going to have the haploid number of chromosomes, and then it fuses with the male gamete, which also has the haploid number of chromosomes. They fuse together, they fertilize to form a zygote that has the diploid number. But in this case, there is no need of fertilization, because in the egg cell, we are putting a nucleus from a somatic body cell, which is already diploid. So thus, now the egg cell has become diploid. So that is why we have removed the nucleus and why we inserted a new nucleus from a somatic body cell. The fourth step is going to stimulate the egg cell to divide. Now how is that done? By giving the cell an electric shock. So electric shock is given to the egg cell. Let me write 2N because this is the new egg cell, and this stimulates the egg to divide. Once the egg starts dividing, the egg is going to be implanted into the uterus of the surrogate. Alright, so what do you think the child is going to be? It's going to be a clone of which animal? Obviously, it's going to be a clone of the animal whose genetic information it carries, which is going to be this particular cell. So it's going to be a clone of the organism that supplied the nucleus. So it's going to have the same genetic information and thus it will become a clone. So let's diagrammatically try to understand this entire process once again. So look at this process now. This is the original cell, the egg cell. So what we have done is we have taken out the nucleus and now this is an enucleated egg cell that does not have any genetic information. This is a somatic body cell. And from this we have taken only the nucleus that contains the genetic information, and this becomes a cell with the diploid number. So this is the new egg cell that has the materials of this original egg cell but the nucleus of the somatic body cell, and this gives rise to a group of cells from that same parent which is known as the clone. This will either be inserted into a surrogate mother, for example the sheep, or it can also be developed in the laboratory, in which case we call it a tissue culture. So why do we have these two processes? Because we have different uses for them. For this kind of animal cloning, we can save endangered species. So species that cannot breed on their own, we can help cloning them. And you can also copy certain elite animals. Again, this is a thing of the future, has still not yet been tried successfully a lot of times. So that is known as reproductive cloning, when you put this clone inside a surrogate mother. So this is reproductive cloning, which is the process that we just studied. The second one, where you need a tissue culture, this is known as therapeutic cloning, which is usually used to make medicines or in some therapy. So what are the uses of therapeutic cloning? You can make transplantable cells, and it's used a lot in tissues, and tissues need to be replaced, and it can also help in replacing damaged tissues. So these are the two uses of therapeutic cloning. So that is what happens in reproductive cloning, which is reproductive cloning and therapeutic cloning. They are different only after the cell mass has started to divide. So if it's inserted into the uterus of a surrogate mother, it's reproductive cloning and can help to save the species and copy animals, because the product of reproductive cloning is going to be a cloned animal. Whereas if this mass is just allowed to grow within a laboratory condition, that means within a tissue culture, it is therapeutic cloning and helps to give us products like cells and new tissues. So that is what happens in therapeutic cloning. Now, therapeutic cloning has a lot of people for it and against it. So let's check those points out. So we are still talking about therapeutic cloning right now, some points for it and some points against it. So some people say that it can be used to cure serious diseases or even disabilities. So that is why it is a good point, because we can replace the bad cells with the good cells. And it's a very important part of stem cell research, which can pave the way for future discoveries and some more beneficial technologies that would not have occurred if their use was banned. The third important argument that people raise in favor of cloning is that stem cells are usually taken from embryos that have otherwise stopped developing and they would have died anyway. So unviable embryos, they can give rise to stem cells, that means these clones. They may not have developed into proper animals, so that is why the stem cells were taken. And the cells are taken at a stage where the embryo does not have a nervous system. So the issue of pain is not related to this topic. So the organism does not feel any pain. We are talking about the clone. It does not feel any pain. Now, what are the arguments against it? It involves the creation and destruction of embryos. So there's a point that at what point do we afford the right to life? The second is that embryonic stem cells are capable of continued division, and then it may develop into cancers as well, cancerous tumors. The third important point against cloning is that more embryos are produced than are needed, so the excess embryos are going to be killed off. So that is one negative point. And the fourth negative point is that with additional cost and effort, there can be alternative technologies that will fulfill the same role. So people are divided on the issue of cloning, and usually they are not able to choose one particular point because of these arguments in favor of and against cloning. So that is all that you need to know about cloning.